Hi, my name is Yenmi, and I'm a pharmacist, and welcome to my YouTube channel, Prescription for Medication Education. This is actually my very first video, and I hope you're as excited about it as I am. I started this channel in the hopes to expand my own knowledge as a pharmacist, and hopefully yours, the viewer, as well. But without further ado, let's begin with my very first video, From Substance to Medication. Okay, so the objectives are to define the term medication, review the brief history of the Food, Drug, and Cosmetic Act, describe the drug development process, learn about controlled substance categorization, and understand the differences between brand and generics. Okay, so defining a medication. First, let's start off with defining what the FDA considers a drug. A drug is any substance that's listed in the United States Pharmacopoeia, National Formulary, or Homeopathic Pharmacopoeia of the United States. These are substances that are intended for diagnosis, cure, mitigation, treatment, or prevention of diseases or conditions. They can be non-food substances affecting structure or function, such as alcohol or tobacco. A medication is also a drug, but it also has some other standards that have to be met. They require strict quality standards during their production process. They contain excipients to help stabilize the medication, improve the efficacy and their safety. And so essentially, every medication is a drug, but not every drug is a medication. For instance, heroin. The FDA also has another category known as biologics. These are also substances that are intended for prevention, treatment, and curing of human conditions, with the exception that these substances come from human, animal, or microorganisms. We get these from viruses, toxins, blood components. They're often complex mixtures that are difficult to categorize, but they can be made up of sugars, proteins, living components such as cells and tissues. Some examples of biologics include vaccines, insulin, monoclonal antibodies. In 1937, a pharmacist in the US compounded sulfanilamide into an elixir using diethylene glycol as the solvent. Unfortunately, diethylene glycol is toxic to the body. This ended up leading to the deaths of over 100 people. Which led to the passage of the 1938 Food, Drug, and Cosmetic Act. Basically, what this law did was it allowed the FDA, it gave the FDA legal power to make sure that the drugs were actually safe to use. It still didn't require the drugs to be effective. That didn't happen too much later on, but it at least required that these medications, before being given out to the population, were shown to be safe. Here's an image of sulfanilamide from the FDA website. An interesting fact about sulfanilamide is that it was actually the first sulfa antibiotic available. The drug development process can be broken down into 5 major steps. The first one being discovering and development, the second one being preclinical research, number 3, clinical research, number 4, FDA review, and number 5, FDA post-market safety monitoring. During the discovery and development process, companies will screen through thousands of compounds of libraries of previously discovered chemicals or organic compounds. New information is often learned about how a disease works, which companies can possibly use as a target for medications to act on. Sometimes companies will also modify active medications, leading to similar types of medications within the same class. Once a company has identified a molecule, they'll label it a lead compound. The company will then file a patent with the FDA either as a composition of patent, which is basically a brand new medication, or a use patent, which is a known drug, but a new use and non-obvious therapeutic use is being identified. The next step is the preclinical research step. This step basically allows for dosing and toxicity. They look at the reproductive effects, any carcinogenic or mutagenic effects, the toxicity for its acute and chronic dosing. This is broken down into two methods. They use the in vitro method, which is basically lining test tubes or petri dishes with human or animal cells and placing the molecule in there and testing for toxicity using that method. The benefit here is that it doesn't harm any animals or humans, but the downside is that it has a low predictive value of toxicity versus the in vivo method, where they basically give the new compound to live animals. Unfortunately, this often involves a large number of animals, but the positive side to this method is that it has a better correlation to finding toxicity data versus the in vitro method. Once the company has finished their preclinical step, they'll move on to the clinical research, which involves humans. So here the company will file a investigational new drug application with the FDA. This is broken down into 3 phases. Phase 1 consists of anywhere between 20 to 100 healthy patients. There are exceptions where they don't use healthy patients where they believe the compound may have severe toxicity. In this case, they'll actually use people with the actual conditions. But basically, phase 1 is just the main idea here is where they try to gather safety and dosage information. Only about 70% of drugs go on to the next phase. In phase 2, this involves a little bit under 100 people. And in this group, they usually gather patients who have the actual condition that we're trying to treat. This phase tests for efficacy, and about 33% of drugs go on to the next phase. And then during phase 3, they'll grab a much larger set of participants, anywhere between 300 to 3,000 volunteers with the condition. Here, we're just looking at efficacy and safety over a much longer duration, and about 25% of drugs go on to the next step. Once the clinical research has been conducted, the drug company will then file what's called a new drug application with the FDA. This contains all the information that was conducted during the clinical research portion. This contains patient information, drug abuse information, directions for use, safety information. The FDA has 6 to 10 months to evaluate the data. If the FDA finds the medication to be safe and effective, then they'll approve it. The FDA will then work with the drug developer to refine the prescribing information, and this is what's termed as labeling. The last and final step is the FDA post-market safety monitoring, most commonly referred to as phase 4. Healthcare providers are prescribing the medication, pharmacists are dispensing the medication. The purpose of this phase is to allow for continued safety monitoring. This is where most of the rare side effects are discovered. Healthcare providers are encouraged to report side effects via the MedWatch program. When companies develop the medications, when they initially file their patent, they're given 20 years for the life of the patent. But in truth, because the drug development process is so long, they usually really only have about 7 to 12 years on average of where they actually have exclusivity of the patent once it's already out in the market. Unfortunately, some substances have the potential for misuse resulting in substance use disorder. In 1970, the Controlled Substance Act was passed, establishing 5 drug schedules, Schedule 1 through Schedule 5. Schedule 1 drugs possess the highest potential for use disorder and misuse and have no medical use and are illicit street drugs such as heroin. Schedule 5 are the least likely to be misused and have very limited physical or psychological dependence, such as Lyrica. The scheduling occurs during the FDA review process, which, if you remember, was step number 4. So basically, the FDA makes a recommendation to the DEA. The DEA then publishes this information in the Federal Register, which allows for the public to give their feedback. And then finally, the DEA finalizes the schedule before the drug is approved. The last topic I want to cover is brand versus generic. So after a brand named drug's patent expires, any drug company can file what's called an abbreviated new drug application. This allows drug manufacturers to not have to conduct clinical trials, but they do have to show bioequivalence. Basically, the generic drug manufacturer has to show that the area under the curve and peak concentrations fall within 80 to 125% of the original brand's value. So for instance, if the original brand name's peak levels were 100 milligrams per ml, then the generic should have a minimum of 80 milligrams per ml and a maximum of 125 milligrams per ml. Both the brand and the generic will be listed in the FDA's approved drug products with therapeutic equivalence evaluations, also known as the Orange Book. Generics must have the same active pharmaceutical ingredient, dose strength route, but they can have some differences such as the inactive ingredients, the shape, and the color. Thank you for joining me on my very first video on this channel. I hope you enjoyed it. Please subscribe and leave me any feedback. I would greatly appreciate it. For my next topic, I'll likely cover dosage form and administration routes. I also have references on here as well for those of you who are interested. Once again, thank you so much. Have a great day.